Now I am going to discuss the respiration, both in plants and animals actually. It's a cellular respiration. Yesterday I was talking about the different steps involved in respiration. For example, there is breathing, external respiration, transportation of the gases, internal respiration, and cellular respiration. Actually, this is a cellular respiration. It occurs both in plants and animals, right? Respiration usually occurs in all the living organisms. And based on the presence or absence of the oxygen, that respiration can be classified into two types. One is the aerobic respiration, and another one is the anaerobic respiration. Respiration occurs in presence of oxygen. Respiration occurs in presence of oxygen is called as the aerobic respiration. Respiration occurs in the absence of oxygen is called as the anaerobic respiration. Right? I think you know these things. Usually this anaerobic respiration occurs in prokaryotes like bacteria. Right? Aerobic respiration occurs in all the living organisms, both in plants and animals. Right? And that aerobic respiration actually occurs in mitochondria. You know that mitochondria are one of the organelles in all the eukaryotic cells. Okay? Of course, they are present in some cases, in some others also. You see, as I told you yesterday, for example, an eukaryotic cell consisting of nucleus, endoplasmic reticulum, Golgi, ribosome, lysosome, mitochondria, chloroplast, etc., etc. Okay. So actual respiration, actual aerobic respiration occurs in mitochondria. So that before studying the actual respiration in the cell, you have to know the structure of the mitochondria. Again, that mitochondria is also a double membranous structure. So it has two membranes. This is the outer membrane, for example, outer membrane. This is the inner membrane. This is inner membrane, right? This is outer membrane. This is inner membrane. And this inner membrane is extended into finger-like structures like this. Into finger-like structures. Okay, these finger-like structures can be called as the crista, singular, cristae, plural, right? And these finger-like cristae are extended into the stroma of the mitochondria. And on each crista, we can see many button-like structures like this, and these button-like structures are called as the Racker's particles, Racker's particles, or F0, F1 particles. Right? So these Racker's particles having the system electron transport system, or you can say electron transport chain, where actually the aerobic respiration occurs. Okay? So in addition to these different parts, this mitochondria may have some ribosomes also, to some extent some DNA also. Okay? Anyway, this is a brief information about the structure of the mitochondria, where actually the aerobic respiration occurs. Right? Okay, so come to this aerobic respiration. Respiration occurs in presence of oxygen. So before that, I can say according to your syllabus, that aerobic respiration or anaerobic respiration consisting of different steps. For example, in anaerobic respiration, there are two steps. One is the glycolysis, and another one is the fermentation. So anaerobic respiration consisting of these two steps, glycolysis and fermentation. Aerobic respiration consisting of three steps. One is the glycolysis. Second one is the Krebs cycle. And the third one is the terminal oxidation, or you can say oxidative phosphorylation. So these are the three steps present in aerobic respiration. So as you are seeing here, glycolysis is a common process, first step both in aerobic and anaerobic respiration. Let us study that glycolysis. Okay? Glycolysis. Okay. Glycolysis. It's a combination of two words, glyco and lysis. Glyco means glucose. Lysis means breakdown. The process of breaking down of the glucose molecule. The process of breaking down of this glucose molecule into carbon dioxide and water is called glycolysis. This is the definition of that term. The process of breaking down of the glucose molecule into carbon dioxide and water is called glycolysis. If this glycolysis occurs in presence of oxygen, it is called aerobic respiration. If the same glycolysis occurs in the absence of oxygen, it is called anaerobic respiration. Right? Let us study in detail that glycolysis. For 5 marks, you have to study this glycolysis. Okay? This glycolysis actually occurs in the cytoplasm of the cell. Remember that. About 1 mark questions. The location of the glycolysis. Glycolysis occurs in the cytoplasm of the cell. Okay. So I told you this is the breaking down of the glucose molecule. Where to get that glucose molecule? That glucose molecule is transported into that cell through that blood circulation, and that glucose is produced in the digestive system, in the small intestine. It's a different aspect. When we take the food, that food will be broken down. Suppose when we take the carbohydrate food, that carbohydrate, all the carbohydrates are digested ultimately, okay, into glucose. So that glucose is the end product of the carbohydrates digestion. So from that small intestine, through that blood circulation, this glucose will be transported to all the body cells, where actually that glucose will be broken down. That process is called as the glycolysis. Right? Here, concerned with the glycolysis, we can define still to specify the meaning of the glycolysis, the process of breaking down of the glucose into pyruvates can be called as the glycolysis. In general, I wrote that equation. That equation can be applied even in general for respiration. Breaking down of the glucose molecule into carbon dioxide and water can be called as a respiration. In that process, that glucose is broken down. Carbon dioxide and water will be released. Of course, in addition to that, energy is also liberated. So it is a general equation. It's a glycolysis actually, but for the respiration, we can define that term. But specifically, what is the meaning of glycolysis? Specific meaning, the process of breaking down of the glucose molecule into pyruvates can be called as glycolysis. That equation is applicable to both glycolysis and in general respiration. Specifically for glycolysis, the process of breaking down of the glucose molecule into pyruvates is called glycolysis. right? So I told you it occurs in the cytoplasm of the cell. Let us study the different steps. Okay, after completing the glycolysis, we can get 8 ATP. What is the net gain of glycolysis? If the question is asked, so 8 ATP. Let us study how we can get 8 ATP. Okay, glycolysis, actually that glycolysis process can also be called as the EMP pathway. EMP pathway. Ernest, Mayerhoff and Parnas. These are the three different scientists who worked together and studied the glycolysis in detail. So that glycolysis process can also be called as the EMP pathway. Okay, and this glycolysis process consisting of different steps. There may be 7 to 8 such steps are there. At the end of that, we can get 8 ATP and 2 pyravates. And 2 pyravates. Each pyruvate is a 3 carbon compound. If we get both this 8 ATP and 2 pyravates, that process is correctly studied. Okay, let us study that. Glucose is there. This glucose, the first step of the reaction actually is first phosphorylation. First phosphorylation. It's the first step in the glycolysis process. In the glycolysis process, first phosphorylation is the first step, during which that glucose combines with the ATP, to produce glucose 6-phosphate. This is the first step present there. So the first step is the first phosphorylation, during which this glucose is phosphorylated in presence of ATP. This will become glucose 6-phosphate. It is called 6-phosphate because here one phosphate molecule is attached to the 6th carbon of the glucose. This is the meaning. Glucose 6-phosphate means here one phosphate is there. That phosphate is attached to the 6th carbon of this glucose. So it is called glucose 6-phosphate. So this ATP will become ADP. Right? This is first step in glycolysis process. Remember that in this first step, we consume one ATP molecule. So after getting so many number of ATPs, we have to deduct the number of ATPs which are consumed into the reaction. Okay, now one ATP is consumed, we have to deduct that from total. Okay, first reaction. Second reaction is the isomerization. Isomerization. In this step, this glucose 6-phosphate will be isomerized into fructose 6-phosphate. Fructose 6-phosphate. Glucose will be converted into fructose. So that glucose 6-phosphate will be isomerized into fructose 6-phosphate. You know that both glucose and fructose are the isomers. The same formula, C6H12O6. Okay, so both of them are isomers. So this is the isomerization. Glucose 6-phosphate is isomerized into fructose 6-phosphate. Third step is the second phosphorylation. Second phosphorylation, during which this fructose 6-phosphate will be phosphorylated once again in presence of ATP. Remember that here also one more ATP is consumed into the reaction. This fructose 6-phosphate will become fructose 1,6-diphosphate, DP, diphosphate. In this fructose now there are 2 phosphates, right? Out of that 2, one phosphate is attached to the first carbon and another phosphate is attached to the 6th carbon. So this is 1,6-diphosphate, fructose 1,6-diphosphate, right? Third reaction is over. After that, the third reaction, we consume 2 ATP. Okay, 2 ATPs. So third reaction is over. Step can be called as the cleavage. That fourth step can be called as the cleavage. During that reaction, this fructose 1,6 diphosphate will be cleaved into DHAP, and PGALD. DHAP and PGALD. Okay, from hereafter that there are this PGALD and DHAP, both of them are isomers. Here what happens? This DHAP will be isomerized into PGALD. Will be isomerized into PGALD. So here at the end of this fourth reaction, we can get 2 PGALD. Each PGALD is a 3 carbon compound. Phosphoglyceraldehyde. This is dihydroxyacetone phosphate. At this moment, this DHAP will also be isomerized into PGALD. Right? Now, at this moment, we have 2 PGALD. Here afterwards, each PGALD enters into a separate chain of reactions. Okay? Let us study that separate step. So, the fifth reaction, fifth step is actually dehydrogenation. Dehydrogenation. Right? Dehydrogenation. Removal of hydrogen is called dehydrogenation. Here, there is a PGALD. It's a 3 carbon compound. Okay? 3 carbon compound. It undergoes a dehydrogenation, and dehydrogenation occurs in presence of NAD. Nicotinamide adenine dinucleotide. Okay? And this PGALD will become 3-PGA. And this NAD becomes NADH2. 1 NADH2 is equal to 3 ATP. I told you, after the fourth step, we got 2 PGALD. Now we are writing only 1 PGALD. Another PGALD also entering into the same reaction, so that at the end of this particular step, we got 2 NADH2 and 2 3-PGA. 1 from each PGALD. Okay? So 3-PGA and NADH2. 1 NADH is equal to 3 ATP. Next step is the, after this next step is the dephosphorylation. Next step is the dephosphorylation. At this step, that 3-PGA, actually the 1,3-dipga, it is 1,3-dipga. To explain in detail, okay, 1,3-dipga. Now this 1,3-dipga undergoes dephosphorylation, to become 3-PGA. Here 1 ADP is there. Okay, 1,3-dipga plus ADP. Okay, it gives rise to 3-PGA plus ATP. So here 1 ATP is produced. In that, one more PGALD is there, from which also another ATP is produced. So at the end of this reaction, we got 2 NADH2 and 2 ATP. 2 NADH2 and 2 ATP. Okay. Next step is the isomerization. Next step is the isomerization, during which that 3-PGA, remember that this PGA, phosphoglyceric acid, is a 3 carbon compound. So this is also 3 carbon compound, right? This 3-PGA will be isomerized into 2-PGA, 3 carbon compound. So both 3-PGA and 2-PGA are isomers, right? Next step is the dehydration. Next step is the dehydration. What do you mean by dehydration? Removal of water is called dehydration. Okay, dehydration. At this step, that 2-PGA 2-PGA, 3-carbon compound, undergoes dehydration. So there is a removal of water. So it will become phosphoenol pyruvate and water is liberated. Phosphoenol pyruvate is also a 3-carbon compound. Right? The last step is the dephosphorylation. Dephosphorylation. So at this step, one phosphate is here, that phosphate is removed. Again, that phosphate is accepted by that ADP. So here, phosphoenol pyruvate undergoes dephosphorylation in presence of ADP. It will become pyruvate. Pyruvate. ADP becomes ATP. So at this moment also, after this reaction also, we got 2 ATPs. 2 NADH2, 4 ATP. 1 NADH2 is equal to 3 ATPs. So there are 2 NADH2, 6 ATP plus 4 ATP. Totally 10 ATPs are produced. But out of the 10, you know that in the beginning we consumed 2 ATPs from outside. We have to deduct that 2. So 10 minus 2 is equal to 8 ATP. So 8 ATP, the net gain of glycolysis is 8 ATP. This is the glycolysis process. At the end of this glycolysis, we got 2 pyruvates with 3 carbons each. 2 NADH2, 4 ATP. But in the beginning, we utilized 2 ATP. That's why we have to deduct. So ultimately, as I told you, 1 NADH2 is equal to 3 ATP. So that there are 2 NADH2. So is equal to 6 ATP plus 4, 10 ATP minus 2, 8 ATP. So the net gain of glycolysis is 8 ATP. And the products obtained are the 2 pyruvates. In addition to that, 8 ATP. This is glycolysis. Okay? But in the exam, you have to write the glycolysis chain, not these equations or not this reaction. Directly you have to write the chain. Let me write that chain here. Okay? The same steps. It is not present in your textbook. Remember that. But I explained to convince you. Okay? Now let me write that chain. Glucose. It's a 6-carbon compound. It undergoes, in the first step, it undergoes phosphorylation. It undergoes phosphorylation. Phosphorylation means addition of phosphate. Addition of phosphate. So that one ATP is consumed into this reaction. ATP becomes ADP. Glucose becomes glucose 6-phosphate. It's also a 6-carbon compound. First reaction is over. This is the first phosphorylation. You have to write the arrow mark correctly. ATP gives rise to ADP. It means from that ATP, adenosine triphosphate, one phosphate is removed. That phosphate is accepted by this glucose, so that this glucose becomes glucose 6-phosphate. Right? First reaction is over. Second reaction, isomerization. I told you, this glucose 6-phosphate is isomerized into fructose 6-phosphate. Second reaction is over. Third reaction is second phosphorylation. Now this fructose 6-phosphate is phosphorylated again. So in this step also, one more ATP is utilized. That ATP becomes ADP. One phosphate is removed from this ATP. That phosphate is accepted by this fructose 6-phosphate. So this fructose 6-phosphate becomes fructose 1,6-diphosphate. It's a 6-carbon compound with 2 phosphates, and that 2 phosphates are present in this glucose. The first phosphate present in the first carbon and another phosphate present at the 6th carbon. This fructose fructose 1,6 diphosphate. Next step is cleavage. Fourth step, cleavage. Cleavage means division. At this particular step, this fructose 1,6 diphosphate 6 carbon compound will be cleaved, will be divided into two compounds with 3 carbons each. That two compounds are, one is the PGALD, phosphoglyceraldehyde, 3 carbon compound. Another compound is the DHAP, dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Okay, and I told you both this PGILD and DHAP are isomers. This can be isomerized into this one, or this can be isomerized into this one. But here this DHAP will be isomerized into PGILD. So at the end of this fourth reaction, we can get 2 PGILD. And each PGILD enters into a separate chain of reactions. A separate chain of reactions. So here afterwards, we have to multiply the product into 2, because there are 2 PGALD. Each PGALD enters a separate chain of reactions. Let me draw one chain. Okay? Another chain is understood. We have to multiply into 2. Okay? The next step, you have to continue the chain. Okay? You have to continue this chain, PGALD. PGALD. Okay, it's a 3 carbon compound. The next step is the dehydrogenation. One hydrogen molecule is removed from this compound. Okay, one hydrogen molecule is removed from this compound. That hydrogen molecule is accepted by NAD. It becomes NADH2 or NADPH2. I told you one NADH2 is equal to 3 ATP. Okay, one PGALD gives rise to one NADH2. One more PGALD is there, it also can give another NADH2. So at the end of this reaction, we can get 2 NADH2. One NADH2 is equal to 3 ATP. So that we can get 6 ATPs. Right. So this PGALD will become, after removal of the hydrogen, this will become 1,3-dipga, 3 carbon compound. Number of carbons will be continued. Okay, 1,3-dipga. The next step is the dephosphorylation. Dephosphorylation. From this phosphoglyceric acid, one phosphate is removed. Okay, that phosphate will be accepted by ADP. That ADP becomes ATP. So here we got one ATP. One more chain is there. There also we can get another ATP. So at the end of this reaction, we got 2 ATPs. This 1,3-dipga, after removal of one phosphate present in the first carbon, this will become 3-PGA, 3-phosphoglyceric acid. Okay, 3 carbon compound. This 3 is the position of the carbon. To the third carbon, that phosphate is attached. When phosphate is there, that phosphate is attached with third carbon. Okay, so this is 3-PGA. Right, 3-PGA. Next reaction is this 3-PGA is isomerized into 2-PGA. There is isomerization. 3-PGA gives rise to 2-PGA. 3 carbon compound. Isomerization. So next step is the dehydration. Removal of water. From this 2-PGA, one water is removed out of the reaction. This will become, this 2-PGA becomes phosphoenol pyruvate, 3 carbon compound. Okay, last reaction, dephosphorylation. Dephosphorylation. Again, one phosphate is removed from this PEP, phosphoenol pyruvate. Okay, I will write it here. Phosphoenol pyruvate. It undergoes dephosphorylation. One phosphate is removed. That phosphate is accepted by ADP. It will become ATP. This compound will become pyruvate. This compound can be called as a pyruvate or pyruvic acid. It's a 3 carbon compound. And it is the end product of glycolysis. So at the end of glycolysis, we can get 2 pyruvates with 3 carbons each. We can get totally 10 ATP, but we have to minus 2 ATPs. So that 8 ATP is the net gain of glycolysis. So at the end of the glycolysis, we can get 2 pyruvates, we can get 2 NADS2, we can get 4 ATP, but we have to deduct 2 ATP, so that the net gain of glycolysis is 8 ATP, right? And the compounds obtained is the pyruvate. Totally 2 pyruvates are obtained. Each pyruvate is with 3 carbons each. This is glycolysis. It occurs, this glycolysis occurs in the cytoplasm of the cell. In the cytoplasm of cell. So glycolysis is over. Right. If we talk about the aerobic respiration, this glycolysis enters into another step. It is the Krebs cycle. It is the Krebs cycle. Krebs cycle, right? And this particular step was well studied by Hans Krebs. That's why this name is given, Krebs cycle. It is also called as the citric acid cycle, because citric acid or citrate is the first stable compound obtained in this particular cycle. So it is called the Krebs cycle, or it's also called as the citric acid cycle. Okay? Citrate cycle, like that. Let us study the Krebs cycle. Now, both the pyruvates which are produced during glycolysis in the cytoplasm will be transferred into the stroma of the mitochondria. Into the stroma of the mitochondria. If this is mitochondria, for example, this is the stroma of the mitochondria. This is a cell, for example. This is mitochondria. In the cytoplasm of this cell, glycolysis occurs. 2 pyruvates are produced. That 2 pyruvates are transferred into the stroma of the mitochondria. Both the pyruvates are entered into the stroma of the mitochondria. Now, each pyruvate enters into a separate cycle. It is the Krebs cycle. So there are 2 Krebs cycles in aerobic respiration. We study one Krebs cycle. Another cycle is understood. We have to multiply that product into 2. Okay? Right. So Krebs cycle occurs in the stroma of the mitochondria. Remember that. Okay? Let us study this particular step. Okay? Okay. Let us study the Krebs cycle. Hans Krebs. Okay? I told you it is also called as the citric acid cycle or citrate cycle. You see, pyruvate is the end product of glycolysis. This pyruvate enters into Krebs cycle. Okay? This pyruvate is a 3-carbon compound. Now, this pyruvate undergoes both dehydrogenation and decarboxylation. Decarboxylation means removal of carbon dioxide. Dehydrogenation means removal of hydrogen. That hydrogen is accepted by NAD. It will become NADH2. Because of the removal of one carbon, this 3-carbon compound becomes a 2-carbon compound. It is the acetyl coenzyme A. Acetyl coenzyme A. This is the first reaction occurs in the Krebs cycle. And this particular reaction occurs on the membrane of mitochondria. Remember that. Okay? So, a 2-carbon compound is obtained. This 2-carbon compound, acetyl-CoA, combines with a 4-carbon compound. It is the oxaloacetic acid. Oxaloacetic acid. Oxaloacetic acid. It's a 4-carbon compound, and this oxaloacetic acid is present in the stroma of the mitochondria. So there is a reaction between this 4-carbon compound oxaloacetic acid plus acetyl coenzyme A 6-carbon compound is produced. It is the citrate or citric acid. You can write citric acid or citrate. This is a 4-carbon compound. This is a 2-carbon compound. So that a 6-carbon compound is obtained. It is the citric acid or citrate. It is the first stable compound obtained in this Krebs cycle. Hence, this cycle is also called as the citric acid cycle. And this particular reaction occurs in presence of an enzyme. It is the citrate synthetase. Citrate synthase or synthetase. Okay? Synthase or synthetase. So, this reaction is over. Actually, this particular reaction and here afterwards, all that reactions occur at the center of the stroma of the mitochondria, right? So, by this time we got one NADH2 in each Krebs cycle. There are 2 Krebs cycles, so that we got 2 NADH2, 2 CO2, right? Now, this citrate undergoes, or citric acid undergoes again both decarboxylation, removal of carbon dioxide, and dehydrogenation, removal of hydrogen. Again, that hydrogen is accepted by NAD, that will become NADH2. Okay? So again, 1 carbon compound is removed. This 6-carbon compound, citric acid, will become 5-carbon compound. That 5-carbon compound is the alpha keto glutaric acid. Alpha keto glutaric acid is a 5-carbon compound. Alpha keto glutarate, you can write 5-carbon compound. Again, that alpha keto glutaric acid again undergoes decarboxylation and dehydrogenation. Okay, again that hydrogen is accepted by NAD, that will become NADH2. It will become a 4-carbon compound. That 4-carbon compound is the succinic acid. Succinic acid or succinate. It's a 4-carbon compound. By this time, you see here 1, 2, 3 NADH2 are produced. 3 carbon dioxide are also liberated. Now this succinic acid undergoes dehydrogenation. Succinic acid undergoes dehydrogenation, removal of hydrogen. But here that hydrogen is not accepted by NAD but accepted by FAD. That will become FADH2. FADH2, flavin adenine dinucleotide. FAD means one FADH2 is equal to 2 ATP. Remember that. Now this compound obtained is the malic acid. That malic acid undergoes dehydrogenation. It will become oxaloacetic acid. So there is a dehydrogenation. So that hydrogen is accepted by once again NAD. That will become NADH2. Thus cycle is repeated. Now you can see here that one citric acid cycle, or you can say one Krebs cycle, can produce 4 NADH2. 1, 2, 3, 4. 4 NADH2 and 1 FADH2. I told you there are 2 Krebs cycles. So totally there are 8 NADH2, 2 FADH2, and one more here, one phosphate is removed. That phosphate is accepted by GDP. That will become GTP. 1 GTP is equal to 1 ATP. So at the end of each Krebs cycle, we can get 4 NADH2, 1 FADH2 and 1 GTP. 4 NADH2, 1 NAD is equal to 3 ATP. 1 FADH2 is equal to 2 ATP. 1 GTP is equal to 1 ATP. As there are 2 Krebs cycles, totally 8 NADH2 are produced. 8 into 3, so 24 ATPs. Okay, 24 ATPs. Right. 2 FADH2 are produced. 1 FADH2 is equal to 2 ATP. Okay, there are 2, this one, 2 ATP. So that 4 ATPs are produced. There are 2 GTPs are produced. 1 GTP is equal to 1 ATP. So that 2 ATPs are produced. Totally 30 ATPs are produced in the Krebs cycle. What is the net gain of Krebs cycle? If the question is asked, so 30 ATP, including both the Krebs cycles, because both the Krebs cycles are the Krebs cycles of one glycolysis, one glucose molecule, right? So what is the net gain of Krebs cycle? If the question is asked, so 30 ATP. Sometimes the question will be asked, what is the net gain of each Krebs cycle? The net gain of each Krebs cycle is 15 ATP. Out of that 15 ATP, 12 ATP from 4 NADH2, 2 ATP from 1 FADH2, 1 ATP from 1 GTP. Totally 15 ATPs are produced from each Krebs cycle. As there are 2 Krebs cycles, totally 30 ATPs are produced. Okay, so the net gain of Krebs cycle is 30 ATP. Net gain of glycolysis is 8 ATP. Totally 38 ATPs are produced. What is the net gain of aerobic respiration? 38 ATP is the net gain of one aerobic respiration. 38 ATP because of breakdown of one glucose molecule. In aerobic respiration, one glucose molecule can produce 38 ATPs totally. Right? So this is information about the Krebs cycle. The third step in the aerobic respiration is the terminal oxidation. Terminal oxidation. It can also be called as the oxidative phosphorylation. Oxidative phosphorylation. Okay, and this third step of the aerobic respiration occurs in the Racker's particles, where the ETS or ETC is present, electron transport system, or you can say electron transport chain. Okay, and this chain is present on the Racker's particles of the mitochondria. I told about the Racker's particles. If this is the mitochondria, this outer membrane, this is inner membrane, this is the crista, I told. Okay. Okay, on this each crista there are some button like structures. These are called as the Racker's particles. On this Racker's particles, this system is present, ETS. Okay, and the last step of the aerobic respiration occurs on this ETS, electron transport system. That electron transport system consisting of different compounds. For example, at the center there is a compound called as the ubiquinone. Okay, to that ubiquinone on one side NADH2 is attached, on the other side FADH2 is attached. From this ubiquinone, the next compound is the cytochrome B. Next one is the cytochrome B. Next one is the cytochrome F. Next one is the cytochrome A. That leads into cytochrome A3. This is a chain. This chain can be called as the electron transport chain. It is called so because electrons are transporting. Electrons are moving from one compound into another compound. While they are moving, actually they are carrying the energy, and by utilizing that energy, that NADH2, that FADH2, ultimately I told you, NADH2 can give rise to 3 ATP, FADH2 can give rise to 2 ATP. So on this chain, while the electrons are moving with energy, that ATPs are produced. Okay, let us study that. So NADS2, when there is the removal of the hydrogen. Because of the removal of hydrogen, that hydrogen thus removed is splitted into protons and electrons, and that electrons carry the energy. Protons, for example, hydrogen is a proton, and these protons are moving all along the length of the chain, like this. Okay, these electrons with energy come to this ubiquinone, and from that ubiquinone, while that electrons are moving from ubiquinone to cytochrome B, a part of the energy is utilized to produce 1 ATP. So there is ADP plus inorganic phosphate gives rise to ATP. A part of the energy present in this electron is utilized to produce 1 ATP. So when one NADS2 is broken down, first ATP is produced at this particular location while the electrons are moving from ubiquinone to cytochrome B, right? Second ATP is produced while the electrons are moving from cytochrome F, from cytochrome B to cytochrome F, okay? Second ATP is produced as usual. Second ATP is produced at this location. Electrons are moving, and third ATP is produced while the electrons are moving from A to A3. So that when one NADS2 is broken down, 3 ATPs are produced as shown in the figure. Ultimately, that electrons are passed into the O, means half O2 present here. At the same time, the hydrogen molecules, you see these hydrogen protons are moving. Now there is a reaction between the hydrogen and that electrons. So hydrogen molecule is, hydrogen ion means protons and electrons are combined once again. H2 is produced. That H2 combines with half O2 to produce water. Thus, when one NADS2 is broken down, 3 ATPs are produced. Okay, like that. When one FADS2 is broken down, as usual, this hydrogen is removed in presence of that enzyme. That hydrogen will be splitted into protons and electrons. Electrons once again carry the energy. While the electrons of the FADS2 are moving from ubiquinone to cytochrome B, first ATP is not produced. Instead of that much of energy will be converted into heat, and that heat is liberated along with the expiring air. We are feeling that, we can feel that the expiring air having the heat. That heat is nothing but it is the heat produced by the energy present here. So when the electrons of the FADS2 moving from ubiquinone to cytochrome B, a part of the energy, instead of producing the ATP, that part of the energy will be converted into heat, and that heat is liberated out along with the expiring air. That's why at this particular location, when FADS2 is broken down, ATP is not produced. While that electrons are moving from here to here, first ATP is produced. Here, second ATP is produced. Thus, when one FADS2 is broken down, 2 ATPs are produced. As usual, ultimately, water is formed. This is the terminal oxidation. Oxidative phosphorylation. Oxidative phosphorylation. Here, phosphorylation, addition of phosphate to ADP, phosphorylation occurs in presence of oxygen. That's why it is called oxidative phosphorylation. Yesterday I told about the oxidative phosphorylation and the photophosphorylation. Phosphorylation occurs in presence of sunlight, photophosphorylation. Phosphorylation occurs in presence of oxygen, oxidative phosphorylation. Photophosphorylation occurs in presence of sunlight. Oxidative phosphorylation occurs in presence of oxygen. Right? Thus, when one NADS2 is broken down, 3 ATPs are produced. When one FADS2 is broken down, 2 ATPs are produced. This is the oxidative phosphorylation. Okay? So, with this, aerobic respiration is completed. Okay? In aerobic respiration, when one glucose molecule is broken down, totally 38 ATPs are produced. So this is the net gain of aerobic respiration. Actually, the net gain is 36 ATP according to recent investigation. 2 more ATPs are consumed while transferring that pyruvates from cytoplasm into the matrix of the mitochondria. But in your book, it is not given, I think 38 ATPs are given only. Okay. So when one glucose molecule is broken down in aerobic respiration, 38 ATPs are produced. Okay. So aerobic respiration is over. Anaerobic respiration. So I told you, in anaerobic respiration there are 2 steps. One is the glycolysis. Another one is the fermentation. Right? Glycolysis as usual. Okay. And this fermentation process or anaerobic respiration occurs in the absence of oxygen, and it occurs in most of the prokaryotic organisms. So as usual, when this glycolysis occurs, 8 ATPs are produced. Whatever may be the organism, when there is glycolysis, the net gain is 8 ATP. Out of that 8 ATP, 2 NADH2 and 2 ATP. Remember that. Okay. So in case of anaerobic respiration, fermentation is the second step after glycolysis. Fermentation is of 2 types. One is the lactic acid fermentation and another one is the alcoholic fermentation. So there are 2 types of fermentations. One is the lactic acid fermentation and another one is the alcoholic fermentation. Right? During lactic acid fermentation, in this lactic acid fermentation, lactic acid is produced from that pyruvate. You know that pyruvate is the end product of this glycolysis. Okay? There are 2 pyruvates. Remember that. When there are that 2 pyruvates, both the pyruvates when enter into lactic acid fermentation process, and that fermentation occurs in presence of 2 NADH2. Remember that. These 2 NADH2 are produced during glycolysis. Okay. So in presence of these 2 NADH2, these 2 pyruvates are fermented and converted into the lactic acid. So during the conversion of these 2 pyruvates into lactic acid, these 2 NADH2 are utilized, so that the net gain of anaerobic respiration is just 2 ATP. 2 ATP only, because out of that 8 ATP, I told you 2 NADH2, 2 ATP, and that 2 NADH2 are utilized during the conversion of pyruvates into lactic acid. So the net gain of anaerobic respiration is 2 ATP. In case of alcoholic fermentation also, these 2 pyruvates are converted into ethyl alcohol, C2H5OH. Okay, C2H5OH. Ethyl alcohol, of course, carbon dioxide is also liberated. In this case also, during the conversion of the pyruvates into alcohol, again that 2 pyruvates are utilized. So the net gain is only 2 ATP. Whether that fermentation is the alcoholic fermentation or lactic acid fermentation, that 2 ATPs are consumed. So the net gain is only 2 ATP. So this is a brief information about the anaerobic respiration. Right? If the question is asked, write the differences between aerobic and anaerobic respiration, you have to write all these things actually. For example, aerobic respiration. Anaerobic respiration. Okay, this aerobic respiration occurs in all the eukaryotic organisms. That anaerobic respiration occurs in particularly in prokaryotes. Remember that this anaerobic respiration also occurs in the muscles of man also, but during strenuous exercise. While we are doing the heavy exercise to provide the energy fastly anaerobic respiration occurs. As usual, because of that anaerobic respiration, lactic acid is produced. Ultimately, only 2 ATPs are produced. So, anaerobic respiration occurs in all the prokaryotic organisms and even in the muscles of the eukaryotic organisms, but during strenuous exercise. One difference. Aerobic respiration occurs at 3 steps. One is the glycolysis, another one is the Krebs cycle, okay, Krebs cycle, and last one is the terminal oxidation. Terminal oxidation. So there are 3 steps in aerobic respiration. While in case of anaerobic respiration, only 2 steps are there. One is the glycolysis and another one is the fermentation. Another one is the fermentation. Difference. In case of aerobic respiration, the net gain of aerobic respiration is 38 ATP. The net gain of anaerobic respiration is just 2 ATP. These are the main differences between the aerobic and anaerobic respirations. You have to write. Okay? Okay, this is the information about both the aerobic and anaerobic respirations. Right. So, in the first PUC syllabus in biology, there are different chapters are there. I told you there are 5 units and each unit consisting of different chapters. Out of so many that chapters, you have studied some of the chapters present in first PU in the SSLC also. So I hope this respiration chapter is also there in your previous chapter. Right. Students, so I am explaining the chapters which are connected with the SSLC chapters only. If I explain the new chapter, you cannot understand. Okay, you will be afraid of that. That's why that chapters are not explained. But no such difficult chapters. All the chapters are very easy. But these chapters, what I explained, are present in your previous classes also. That's why this is a bridge course which is connecting both the SLC and the PUC syllabus. That's why I'm explaining such chapters only. Okay? Right. Now, I think you have studied the phytohormones also. I think it's not concerned with this chapter. In general, I am telling you. You might have studied the phytohormones. You have studied the hormones in general. Okay? Hormones in general. Phytohormones in particular. You know what are hormones? I am giving this information in general, not concerned with any particular chapter. Okay? It's a bridge course. That's why I am explaining that chapters you have studied. You have studied about the hormones. You know what are hormones? Hormones are the chemicals which are secreted. Hormones are the chemicals. Hormones are the biochemicals. Remember that. They are secreted in endocrine glands. In endocrine glands. I told yesterday about the endocrine glands. Okay? Endocrine glands released into bloodstream, concerned with animals, released into bloodstream and reach the target organs. Reach the target organs and function. This is the definition of hormones. Hormones are the biochemicals secreted in endocrine glands, released into bloodstream, reach the target organs where they function. Okay? Simply we can say hormones are the chemical messengers. Hormones are the chemical messengers. Okay, right. I think you have studied different organs and different endocrine glands in your previous class. For example, there are many endocrine glands present in man. Out of that, this is the main gland present there, pituitary gland. Pituitary gland. I think you have studied about this pituitary gland, pituitary hormones. Thyroid gland. Thyroid gland parathyroid gland, parathyroid gland, adrenal glands, adrenal glands, even gonads, etc. are some of the endocrine glands present in the human body. Present in the human body. Right? You have studied about the pituitary gland. And actually this pituitary gland is also called as the master gland. It is called so because it controls the secretion and functions of all other endocrine glands. It is having the control over all these different glands. It is having the control over the secretions of these different glands. So, pituitary gland is also called as the master gland. And that pituitary gland is present at the base of the hypothalamus. You know what is hypothalamus? Hypothalamus is a part of the brain. You have studied about the human brain. Okay? Human brain can be divisible into three parts: forebrain, midbrain, hindbrain. Like that. Forebrain consisting of these parts: cerebrum and diencephalon. Diencephalon. This diencephalon consisting of thalamus and hypothalamus. Hypothalamus. Okay? Below this hypothalamus, this pituitary gland is present. Remember that. Okay? And this pituitary gland secretes so many hormones, and it can store so many hormones secreted by this hypothalamus. For example, this hypothalamus secretes hormones oxytocin. Oxytocin. This can be written like this also. Oxytocin. Okay? Oxytocin. Okay. ADH. Anti-diuretic hormones. This ADH or this oxytocin are actually secreted from this hypothalamus, but stored in a particular chamber of the pituitary gland. In addition to that storage function, this pituitary gland secreting so many hormones. For example, growth hormones, thyroid stimulating hormones, and it is secreting ACTH, adrenocorticotropic hormones. It is secreting gonadotropins like FSH, LH, etc. These are some of the hormones secreted from this pituitary gland, from the anterior part of the pituitary gland, means from the adenohypophysis of this pituitary gland. This is a growth hormone, also called as the somatotropic hormones. It helps in the physical growth of the body. Right? I will explain that in detail while explaining that particular chapter. Thyroid stimulating hormone. As the name indicates, these hormones stimulate the thyroid gland. Because of these hormones, there is a growth of the thyroid gland present here. And the thyroid gland secretes another hormones, thyroxine. So, growth of the thyroid gland and secretion of the thyroxine hormone is controlled over by these hormones secreted from this pituitary gland. ACTH hormones, adrenocorticotropic hormones. And these hormones act upon the glands present at the tip of the kidney, adrenal glands. That adrenal glands present at the tip of the kidneys. Okay? So these hormones acts upon them. So there is a growth of the adrenal glands. In turn, that adrenal glands secrete the hormones like cortical hormones, medullary hormones, etc. because of this ACTH. And these two hormones can be called as the gonadotropins. Gonadotropins. Both FSH and LH are called as the gonadotropins. They are called so because they act upon the gonads. So they are called gonadotropins. Then what are gonads? Sex organs. For example, testes, male sex organ. Ovaries, female sex organ. So they are the gonads upon which these two hormones are acting upon. So that's why these two are called as the gonadotropins. Like, okay, so like that there are different hormones secreted from this pituitary gland. Like the thyroid gland, thyroxine hormone, parathyroxine hormone, adrenal hormones, from these gonads, testosterone hormones, okay, LH hormones, etc. are secreted. So I think you have studied some of these hormones. Okay, of course, you will study that hormones in elaboration in the first PU. Okay, so in plants also there are so many hormones. Hormones present in the plants can be called as the phytohormones. Phytohormones. Plant hormones are called as the phytohormones. And that phytohormones can be divisible into growth promoters and growth inhibitors. Growth promoters and growth inhibitors. I think you have studied, okay? Auxins, for example. Auxins, gibberellins, cytokinins, abscisic acid, ethylene. These are the different hormones which are secreted in the plants. Out of this, these first three are called as the growth promoters because they help in growth of the plant body, growth of the cell, growth of the tissue, growth of the plant in general. Okay. These two hormones are called as the growth inhibitors because they inhibit the growth of that cell or tissue or organ of the plant. Okay. Out of that, this ethylene is also called as the volatile hormones. Volatile hormones. Even ethylene in chemistry you might have studied. It evaporates immediately. So this one also. That's why it is called as the volatile hormones. So these are some of the hormones I think you have studied in your previous classes, but this year you will study the background of the discovery of this hormone, role of this hormone, uses and disuse of hormones, etc. All these hormones you will study the background of discovery, then role of that each hormone, etc. Okay, so let us explain in detail while conducting a class. Right, students, better you understand the glycolysis, Krebs cycle well, because both the glycolysis and Krebs cycle, the questions will be asked for 5 marks. Okay, explain the glycolysis chain, explain the Krebs cycle, or neat labeled the glycolysis chain, Krebs cycle, etc. So, and there are so many 1 mark questions. For example, the number of the net gain of glycolysis is, there will be 4 answers: 8 ATP, 10 ATP, 6 ATP, like that, 2 ATP. The net gain of anaerobic respiration, okay, like that. Net gain of aerobic respiration. In general, net gain of Krebs cycle, there will be 4 answers, like that. So you can observe, when you study that information in detail perfectly, you will be able to answer any type of questions, whether that question is a MCQ or 1 mark question, 5 marks question, CET, NEET, whatever it is. If you know the concept correctly, then you will be in a position to answer any kind of question which will be posted on. Okay, so thank you.
All this time I've been holding on so tight, 'cause all that I do